This particular lecture, there's a few parts of it that are going to be really important and several parts of it that are informational. Straight up, right off the top, I want to tell you I am not going to ask you to label or remember any of the groups. I want you to take away the big ideas. So, how are these things characterized? And, basically, what's a plastid? What might lead to multicellularity? So, there's a lot we're going to talk about. And I want you to focus in not on the stupid little individual differences between the groups so much as the big picture. So protozoans are basically everywhere. Molecular data, which is what I tend to rely on because I'm a geneticist, places these organisms, places protists into every major supergroup, from the fungus to the plants to the animals, all across the board. They're everywhere. So previously we've talked about domains, and we had 3 domains. What were the 3 domains we talked about? We've really gone into depth on 2, but what was it? Eukaryotes, 1. Prokaryotic, and then prokaryotes are divided into 2 groups, which are 2 domains, which are what? Bacteria and archaea. So we've got these supergroups. Those supergroups underneath them you have domains, and underneath the domains fall the kingdoms. And those kingdoms are huge groups of these other organisms. So here we can see the domains of bacteria and archaea, eukaryota, and then eukaryota gets divided up into protozoans, chromista, plantae, fungi, and animalia. And that's this week. Who knows what will happen next week because it's taxonomy. One of these groups is known as Excavata. They are subordinate to the subordinate to the eukaryotes, and they encompass many, many, many protozoans. Subordinate, by the way, means underneath. What characterizes Excavata is a feeding groove. They literally have a long structure within them in which other organisms are pumped in, or nutrients are pumped in. They form vesicles at the bottom, and those vesicles are exposed to lysosomes that break them down. That feeding groove is what makes Excavata Excavata. Many of these feeding grooves have either cilia attached to them that pump in nutrients, or flagella that whip them in. So up here, you can see, obviously, it looks like a giant mouth at the top. There are some cilia there that are beating, that draw food in to create these vacuoles inside. Excavates undergo what's called phagocytosis. Phagocytosis is the term you will need to know. 100% you will need this one. Phagocytosis occurs where a food particle gets engulfed by a membrane to form a vacuole. This process is exactly like endocytosis from Bio 101. The only difference is it is for sure a food particle coming in. So you're bringing in a food particle. Eventually, lysosomes will then bind to the vacuole, dropping off their acid hydrolases in order to break down the food into its constituent components and recover energy and organic molecules. It is phagocytosis that likely led the way to endosymbiosis. Endosymbiosis being where one organism absorbs another, and they work together for mutual benefit rather than being digested. Likely, that's how we got our mitochondria from an aerobic bacterium. We got our chloroplast from a cyanobacteria. The eukaryotic cell absorbed them and began to use them for its own purposes, feeding them energy, feeding them food, and protecting them, while they in turn gave glucose or ATP. Another group are called the Euglenoids. They're characterized not by being green. Euglena, you typically think of Euglena as green. Euglenoids are actually characterized by these protein layers underneath their plasma membrane, making them super flexible. They can like really contort themselves like other creatures can't, other protists can't. Not all euglena are euglenoids or euglena. They most don't have photosynthetic capabilities. That is, they cannot use sunlight to create their own food. What they do have are plastids, organelles that can synthesize new components. Chloroplasts are a type of plastid. There are many types of plastids. Whenever you see plast at the end of something, it's usually a plastid. They make new components. They make new macromolecules. Every land plant, the Stromatopilla, have plastids. Every unicellular protist called algae has plastids. All the euglenoids have plastids. Plastids were usually taken in by a cell through endosymbiosis. That has allowed for a lot of horizontal gene transfer. It doesn't necessarily mean that there's a direct relationship between the algae, the protozoans, and that have plastids and the land plants, but there could be. Green algae are one of these. They live basically everywhere, and they are photosynthetic. Green algae can produce algal blooms, as we've talked about in the last lecture. This can lead to consequences such as anoxia among fish living in that region, especially at night. We have the red algae. These red algae are usually multicellular, and they lack flagella. And by multicellular, I don't mean that they are like a complex multicellular organism. I mean they are multiple cells linked together to create complex structures. Yes. Oh, sorry. Red algae produce agar, agar and agarose. That's very heavily used in biology. That's what you're going to find in a Petri plate. And I didn't realize this, but apparently there's a new thing going on in labs where you create Petri dish art. I don't know how that happened. Basically, I expect a graduate student was really bored and did it and then took a picture and then it became a thing. So that's a thing now. People are making art using basically jello and bacteria. But cool. Plastids in red algae are very, very complex because what ends up happening is several rounds of endosymbiosis. You start off with a single cyanobacteria being taken in by a heterotrophic eukaryote, and you have a single complex organism with a plastid in it. Now, what happens next is that particular eukaryotic organism will take in another, or will be taken in by another cell to create a multi-cell level plastid. Let me draw a picture. All right, so let's imagine, if you will, you've got one cell that is going to engulf this other cell using what process? What's the process where it's engulfing it called? Phagocytosis. So it takes it in using phagocytosis, and boop, they pop off their membrane. So now we have a double membrane-bound organelle on the inside. Now it gets trickier, right? Because an even bigger organism comes along. Through phagocytosis, takes in that cell. And now it's encased in a vacuole. You've got a plastid that's got 1, 2, 3, 4 bilayers. Happens again. Creating all of these layers ends up leaving the plastid with many, many, many different what are called intracellular spaces or intracellular spaces. It's in these spaces between where the components are not that you can create gradients, like hydrogen ion gradients, that can power reactions to produce macromolecules. Having all of these organisms with their own unique sets of DNA interacting with each other. So there's one. Here's one. And then you got DNA out here. So having these organisms all with their own unique DNA allows for or provides the opportunity for a lot of horizontal gene transfer. That might pop out and the from one of the components into the cytosol of another component, creating a cohesive genome. Lots and lots of back and forth gene transfer. Makes taxonomy using genetics very, very difficult. So like I said, all organisms in Streptophyta, so the land plants, are going to contain one or more type of plastid. We got our alveolates. Oh, these are organisms that have sac-like structures in their cells called alveoli. These sac-like structures are like little pockets. And I swear, you will never see anybody as excited as like my wife when she realizes her dress has pockets. It is a great feeling, and these organisms have pockets. So these alveoli are these modified pockets. They're great for all sorts of things. They're great for helping float, defense, and increase reproduction. How do they help you float? So if you've got pockets, not you, maybe you. Yes, you. Okay, guys, you've got swim trunks on at some point in your life, right? Have you ever noticed that when you jump in, your pockets like filled with gas and they float up? Yeah, okay. It's kind of, and then you sort of squeeze it out and you're like, bloop, big bubble. Cool. All right, stick with me. That actually can help you float. They act almost like these water wings on really excited kid here. It's you've taken gases, you've put it into these pockets in the alveolates, and it lets them float. And that means they don't have to expend energy to swim. Defense. You can fill your pocket with poison. I know some of you have wanted to do that at some point in your life. Fill your pocket with weapons and just start chucking. They just fill them with I don't know, like ninja throwing stars. And you're like, ha, throw them. And then the thing around you dies. You've dreamed it at some point in your life. There was some situation you were in where like, man, I wish I could just pull something out of my pocket and drop this person dead right in front of me. And if you haven't, well, okay, maybe I'm just a sociopath. I have no idea. Increased reproduction. Alveolates fill their pockets, fill their sacs up with reproductive cells. And then as they're going by other organisms, just spill them out. Now, I don't recommend you do that. Don't fill your cells up with your own reproductive cells and throw them at people. But it works for alveolates because then they can increase the reproductive rates. Stromatopilla, another group. It's not nearly as cool as the alveolates because as much as pockets are awesome, these guys just have hairs. Well, what hairs do on the Stromatopilla is provide increased surface area. So you can see on this flagella, there are a lot of hairs. More surface area means more motion. When you guys, have you guys ever gone on a canoe or had a boat with an oar? You know what I'm talking about, a boat with an oar? Yeah, you can imagine it at least. Okay, you put which side of the oar into water? Not trying to trick you. Yeah, the side that's flat, that's got a really big surface area. What happens if you tried using the other side? You're not going to go anywhere because it doesn't have enough surface area to push against the water. What these hairs act like are oars. They help the organism move. The Phytopular Infestants was a type of fungus-like protist that caused the Irish potato famine. It causes currently billions of dollars in potato crops yearly. Basically, it's a Stromatopilla that when it's moist outside, when it's wet, they can swim from one plant down the plant across moist soil to another plant very, very easily. It lets it spread fast. The brown algae, so kelp is a type of stromatopile. So it may look like a huge plant. It's really just a whole bunch of single cells. Rizzarians. Oh, Rizzarians are scary looking. They have spikes made of cytoplasm. The Rizzarians, they are one of the few protists that can fossilize. Their cells, their cytoplasm can actually fossilize because these spikes are made up of chemicals that will readily get transferred into an inorganic form. When I think of them, I always think of, those really spiky balls that you don't go outside and fall for, you like without shoes. They're like brown. I don't know what kind of plant are those from. Do you guys know? Maple? Yeah, it's like those spiky balls that are on the ground. Japanese maple. Yeah, they suck. That's my whole feeling on them. They remind me of the Rizzarians, just in a much larger form. We use Rizzarian deposits for carbon dating. You don't want to destroy your actual fossil, but you know what? If you destroyed the Rizzarians around that fossil that lived in the same area, you can use it, grind it up, take the carbon, and figure out the carbon on these guys, the ratios in carbon to date the fossil that they're found with. We got our amoebozoans. To go figure, they move through pseudopods. Man, we came up with the word amoeba, and then we used it a ton to describe things. Slime molds are my favorite amoebozoans. I've told you guys before, and I'm going to show you a video in a second. Slime molds are my favorite amoebozoan. They can. I'm going to keep. I want to keep one as a pet. They can navigate mazes. They actually, and super cool. These guys were attracted to the yeast in the beer. They can actually farm bacteria. So the slime molds will grow and create a little corral, and they'll let bacteria grow in the middle. They'll actually pump nutrients to the bacteria to get the bacteria to grow. And then when the bacteria is grown, the slime mold will come in, eat some of the bacteria, and then go back out and start feeding it more nutrients again in order to keep this farm going. They're pretty smart for a single-celled organism. Don't want to say smart. They got some really neat adaptations. And then we got our Epistacons. The Epistacons are the closest related to animals and fungi. Now, it should not surprise you that you are most closely related to fungus as far as groups go, and the connection is the Epistacons. I want you to pay close attention to, you've got what's called a collagen ring right here. That collagen ring is going to pop up in several animal groups. And what's really neat is, and I want to show these side by side, is you've got a single-celled Epistacon here. And these are still single-celled Epistacons, but they're all held together in one space. They've got some adaptations that lend themselves to multicellularity. And you've got your nuclearia, which are going to be soon to be fungus. These are the basal. But as far as the Epistacons go, the choanoflagellate group underneath the Epistacons, that's interesting. They possess genes for cellular adhesion and extracellular communication. Those two genes, write this down, it's going to come up definitely on an assessment. Those two genes, cellular adhesion and extracellular communication, are essential for multicellularity. These organisms need to be able to, in order for you to live, you need to be able to hold all of your cells together, and those cells need to talk back and forth. Does that make sense? If you did not hold your cells together, you would be a puddle on the floor. So, cellular adhesion, hold on to your neighbor, and talk to your neighbor, became vital for survival. And here's why Epistacons are so cool, as far as protists go. They don't just have a convergent evolution. They're not talking about multicellular, getting multicellularity and cellular adhesion because they're adapted to their environment. They have literally the exact same genes we do. The odds of that happening are so remote it's not worth talking about. The exact same gene sequences. This is an example of direct evolution. It made multicellularity easier to achieve. This collagen ring and the structure of the choanoflagellate is the exact same cell that sponges use to get food. It's called a choanocyte cell. Here you have amoebocyte cells. We'll talk about that when we get to sponges. So this is an example of direct evolution. This choanoflagellate is the exact same structure, the exact same genes as that choanocyte cell in a sponge. It's where we came from. So there we go. Protists are hard to classify. But they do have a lot of shared, you do get groups of shared characteristics, like the excavates have that feeding groove. The algae all have plastids. The Epistacons all have multicellularity and cellular adhesion genes. So we'd still try and classify because we are down that fool's errand of taxonomy. Do you guys have questions?